A very big hello to all my wonderful KCET aspirants. I hope that you're doing wonderful, my dear children. And yes, the exam is coming by, but we do not have to be worried about anything because you guys have us with you, right? So today we are going to complete human health and diseases in one shot. Now this video could be around 1.5 hours or 1 hour, something like that. I'll try to make it fast. Remember, this is a one shot video, sort of a revision. So obviously it has to be a bit fast paced, but obviously in understandable manner. I'll try to do so. And I want just one thing from you: have your NCERT with you, have a pen with you. Why? Because there are certain areas which you might feel that it's not very important, we might not get questions on the basis of that, but then I'll be showing you a question somehow. So probably you can just write down, this is something that could be asked. Okay, so I have to probably prioritize this as well in my preparation, or probably sometimes I might tell you that there are certain things which might be a bit extra, okay, which is not completely there in your NCERT, or probably for concept understanding I might tell you about something a bit extra, so you have to just write it down in 1 or 2 words, that's all. Okay, so make sure you have it, and also when you have your NCERT you can relate and learn that okay, ma'am is teaching this thing, I am probably covering this thing, so you feel confident that yes, with this particular video I am going to cover everything that is there in the NCERT. Works? So shall we start? I hope we can start. And my dear children, please be careful that over here we are not getting distracted anywhere else. We are just going to be focused for straight 1 or 1.5 hours. So I hope you're ready. If you're ready, do let me know in the comment section, and let's begin. And also if you have any doubts, ask me. Okay, now dear children, so first thing is, what do you mean by diseases? So there are two types of diseases: infectious and non-infectious. Infectious as in, if I have the disease, then there are higher chances that you can also get the disease if you are near me, or probably there is some exchange of body fluids, or probably I sneeze, or I contaminate the food, whatever. There can be many reasons for the diseases to be transported from one organism to the other organisms. So these type of diseases which can be transferred like this are known as infectious diseases. Now there are some other things which are not contagious. For example, I have it, but there are no chances that you will have it just because you are near me. Okay, just because the food is getting contaminated, just because basically I am sneezing or whatever is there. So these are known as non-infectious diseases. So infectious diseases as in it includes AIDS, or probably the typhoid, or probably pneumonia, all those things. Non-infectious as in cancer. For example, if I have something, there is no chance that you might have it. You may or may not have it, and it depends totally on your body, not because it is going to get transferred. Okay, so that is about infectious and non-infectious diseases. We'll go ahead, we'll talk about certain diseases. Okay, there are different categories. The first category is bacterial diseases, as in these diseases are going to be caused because of bacteria, different bacteria, and the first one among them is typhoid. So typhoid is caused by Salmonella typhi. The name has typhoid. Yeah, it's a bacteria and it causes typhoid fever. Now how do you get typhoid? It is transferred through contaminated food and water. Transferred through what? Contaminated food and water. You know Mary Mallon? Do you know Mary Mallon? So Mary Mallon was a lady who was a cook. Okay, who was a cook. So now this particular person used to go to washroom obviously, and probably might not be washing her hands properly, but with the same hands she used to cook food. And this is how lots and lots of people got infected with typhoid during that particular era. You get it? So you get an idea that this particular disease is being transported from one organism to other organism with the help of contaminated food and water which is there. I hope you get it. Okay, now over here in our body, where does it go and do its effect? So it goes to our small intestine and also our blood and also the other organs, and after they get into our blood and small intestine and everything, what do? There are certain symptoms. So there will be high fever, which is almost around 39 to 40 degrees Celsius. There will be weakness, there will be stomach pain, there will be constipation, headache, loss of appetite as well. Okay, so high fever is something that you should never forget for typhoid. Okay, in typhoid you will be getting high fever, and there will be weakness and stomach pain and everything else, and in severe cases it might lead to intestinal perforation. What do you mean by that? In the intestine there will be perforation, there will be small holes as well, my dear children, and it can lead to death. So mode of infection is contaminated food and water. Please remember it. Okay, it is known as fecal-oral route, as in from the fecus, it will be fecus as in the contaminants that you release from our body, right? So from the fecus, it can get transferred to the food and the water, and if we have it, then we will get diseased, and it's going to affect the small intestine, which is why in severe cases it might lead to intestinal perforation as well. Now there is a diagnosis test for typhoid, and that is Vidal test, my dear children. So in your NCERT there are two diseases for which we have test. One is typhoid, another one is HIV. AIDS. Okay, for AIDS we have ELISA, for typhoid we have Vidal. Please remember that. So these are all the things that you need to remember. High priority thing is intestinal perforation and also high fever caused by Salmonella typhi, and the diagnosis is Vidal test. Cool? Works? So that's about typhoid. Now going ahead with the next bacterial disease, which is pneumonia, my dear children. In pneumonia there are two pathogens given in an NCERT. One is Streptococcus pneumoniae and the other one is Haemophilus influenzae. Haemophilus influenzae, Streptococcus pneumoniae, both of them are bacteria. Now infection site, where does it go and affect? It affects the alveoli in the lungs. You know, in the lungs there are alveoli where maximum exchange of gases and everything takes place. What happens in the case of pneumonia is that this particular alveoli is going to filled with certain fluid, mucus and stuff like that, and what happens when it gets mixed filled with fluid? The exchange of gases would not be taking place. The exchange of gases, which is a normal process, it does not take place, which is why we feel a breathing issue, okay, because no much oxygen is being transported up there. So it affects the alveoli in the lungs, filling them with fluid. It causes breathing issue. Now symptoms are fevers, chills, cough, headache, and in severe cases, please remember all the severe cases you have to remember, lips and the nail, it will turn bluish grey in color. Bluish grey in color. Remember always, pneumonia is going to affect your lungs and everything. If you talk about common cold, it affects mostly your upper respiratory tract, not the lower one. Okay, common cold affects the upper respiratory tract. Pneumonia, it mostly affects the lower respiratory, that is your lungs and everything up there. So you get fever. Okay, you get fever, you get chills, you get cough, you get headache, and in severe cases, lips and nails turn bluish grey in color. Now, transmission, how does it get transmitted? If inhalation of infected, for example, I sneeze. So, obviously, there will be infected droplets over there. Now, if you take in that drop, basically, if you also take in breath like that, or these things, probably I will be putting down saliva as well. Now, if you touch on certain things which has my saliva on it, which has these contaminants, obviously, there are high chances that you can get it. So sharing utensils with an infected person also causes it. Now there are certain other bacterial diseases mentioned in your NCERT. There is nothing else given, okay, about that. That is dysentery, that is plague and diphtheria. So if at all the question comes that dysentery is caused by bacteria, virus, fungi, what is it? So basically it is caused by bacteria. Okay, now there is amoebic dysentery, that is totally different, that is caused by protozoan. This is simple dysentery. Okay, simple dysentery. And now you have plague and also you have diphtheria. Please remember it. Apart from that, there is cholera as well, not chlorella, cholera as well, which is caused by bacteria. So in total we have typhoid, pneumonia, dysentery, plague, diphtheria and also cholera, which is caused by bacteria. Over here the lips and the nails turn bluish grey, that is the severe case, and the infected site or the infection site is what? Alveoli in the lungs. Works? Done? And the two names, please remember them. Now we'll go to protozoan disease, a disease caused by protozoa. So first we have malaria, my dear children, very important one. Okay, so it is caused by Plasmodium. Now Plasmodium, there are different species. Okay, we have P. malariae, P. falciparum and P. vivax. Now please remember, the malignant version of malaria, the most dangerous version of malaria is caused by P. falciparum. Now transmission, how is it transmitted? It is transmitted by the bites of female Anopheles mosquito. So Anopheles mosquito is actually a vector because it carries the pathogen. It is a vector. Why? Because it carries the pathogens and it comes, bites us, and then we get that particular pathogen in our body. So it spreads through bites of the female Anopheles mosquito. Now how is the life cycle happening? So basically I'll tell you, in the female Anopheles mosquito, the Plasmodium which is there, it is in one particular stage. So suppose I am the Anopheles mosquito, okay, and I will be having my salivary glands and everything. In the salivary glands, basically, I would be, it will be a breeding ground for this Plasmodium, which will be in sporozoite stage. So in my salivary gland, the Plasmodium is there in its sporozoite stage. Now I come and bite a human being. When I bite a human being, I am basically inserting a little bit of my saliva. Why? Because in my saliva there are anti-clotting factors as well, so that I can basically drink the blood really peacefully. So whenever there is a clot, I remove that clot with my saliva, and that's how I drink the blood. But when I am doing so, when I am putting my saliva, what is happening? The sporozoite which is there in my mouth, it is also getting inside the body of the organism, that is the human being. So in which stage am I getting affected? If I am a human being, in which stage am I getting affected? In the sporozoite stage. Now what will happen? This sporozoite which is there now in the blood will be moving around, and it reaches the liver. It reaches the liver. In the liver, it is going to affect the cells of the liver, that is the hepatocytes. The cells of the liver are known as hepatocytes. Okay, now it is in the liver affecting the hepatocyte. There it will divide, and the liver cell will burst open. Now it is another stage. We are not going to talk about that stage, but anyways, just remember it is in one particular stage. Okay, now this Plasmodium which is there, my dear children, it will go and attack the next things, that is your RBC, red blood cells. Now this is where things get really bad. So in RBC, my dear children, even though they are dividing, they are also changing their stages, and it reaches a stage which is known as gametocyte stage. What do you call this stage as? I'll write it because it is not there in your NCERT. This is something extra, write it. So gametocyte stage it reaches in the RBC. So when the RBC is now going to burst, there are two things that is going to happen. One is that the Plasmodium is going to be removed in this gametocyte stage. Okay. Second thing that happens is that in RBC, what do we have? We have hemoglobin. So this Plasmodium, it's going to break down this hemoglobin or feed on this hemoglobin, and when it does it, it breaks it into two things, heme and globin. Heme protein and globin protein. Now, the problem is that the heme which is there, it is kind of toxic for the Plasmodium. So, Plasmodium thinks I should be converting into something that is not toxic for me, and that is hemozoan. And that is what? Hemozoan. So, suppose I am the Plasmodium, I am now inside the RBC. Until now, I have not burst open the RBC. Okay, I am inside the RBC, and I am basically going to break down this hemoglobin into heme and globin. Heme is kind of toxic for me. I convert it into something that is known as hemozoan. I convert into something that is known as hemozoan. So I have hemozoan now and the globin molecules which are there. And apart from that, I have also transferred, I have also changed my stage to gametocyte stage. Now what will happen? After a particular point of time, this RBC will burst. When it bursts, the hemozoan is being released, gametocyte is also being released. The hemozoan which is there, it is kind of very toxic for the human body. It is kind of very toxic for the human body, which is why we will be getting chills and high fever. Whenever it is released, we will be getting what? Chills and high fever. Now this is very important symptom of malaria, my dear children. Cool? Works. And what happens? The gametocyte is being released, right? Gametocyte is a stage, okay, gametocyte is a stage where basically these organisms, the Plasmodium which is there, it can form the gametes, and the gametes can fuse and everything. But this does not happen in our body. We just release them in gametocyte stage. Okay, so they are not going to form the gametes in our body. They are in that stage where it can produce but does not produces in our body. Listen to me carefully. Okay, so it is in gametocyte stage. Hemozoan is being released. We are getting chills and high fever and everything. Every 3 to 4 days we get this. Okay, now what happens? Another female Anopheles mosquito comes to us and is there to just drink the blood. But when it drinks the blood, it is going to carry what? The gametocyte which is there in the blood. Now the gametocyte is going to enter the gut of the mosquito, because all the blood is going to the gut of the mosquito. There in the gut of the mosquito, basically these gametocytes are going to form gametes, and then these gametes are going to fuse. Okay, these gametes are going to fuse, that is fertilization is going to happen in the mosquito's gut, and then they are going to form some other stage in the gut itself, and then slowly it is going to get transported back to salivary glands of the mosquito. Now it is in which stage? It is in sporozoite stage, again in the infected stage. So this is how things work. So things that you have to remember is, in what stage does it enter our body? Sporozoite stage. Okay, in what stage does it leave our body? Gametocyte stage. It does not form the gametes. Where does it form the gametes? Inside the mosquito gut. Where does fertilization happen? Inside the mosquito gut. Now back again to the salivary glands, what comes in? Sporozoite. This thing happens again and again and again. Why do you get fever and chills? Because hemozoan which is there, it is being released. When the cell bursts, hemozoan is being released. What are the two cells that are affected by malaria? First thing is liver cells. Then the second camp of malarial cells is what? Malarial parasite is what? The RBC, or what do you call it? Erythrocyte. I hope this is done. Fine. Now, one very important thing, requires two hosts. Okay, it requires two hosts. So Anopheles mosquito is a vector because it carries, and it is also a host because this is where the guys will be what basically undergoing fertilization and everything. So mosquito is acting as a host as well and a vector as well. So human and mosquitoes are host. What is a vector? Just the Anopheles mosquito is a vector as well. I hope this is clear. Please write this. It's very, very, very important. Where does fertilization occur? That is the question that comes. I hope this is done. Clear? Okay, cool. Just go through this picture. This picture is really, really important, and whatever I explained to you is given in this particular picture as well. Chalo, let's go ahead. So now we have the next protozoan disease, which is what? Amoebiasis. In amoebiasis, who causes it? It is Amoeba histolytica. It is what? Amoeba histolytica, which is again a protozoan that was caused by Plasmodium. Who was the one causing the malignant version of malaria? It was Plasmodium falciparum. Okay. Now the disease caused by Amoeba histolytica is amoebiasis. You also call it amoebic dysentery. Remember, just dysentery if it's there, that is a bacterial one. Amoebic dysentery if it is mentioned, amoeba is a protozoan. So this is a protozoan disease. Now symptoms, again you will have constipation, you will have abdominal pain, you will have cramps and blood and mucus in the stool. So whenever the term dysentery comes, that means in the stool there will be blood as well as mucus. That is the term dysentery means. Okay. So constipation, abdominal pain, there will be cramps, and then after all of this thing, after constipation everything, there will be what? Blood and mucus in the stool whenever it is there. Now, how is it transmitted? It is transmitted with contaminated food and water. I told you the fecal matter which is there, which is removed, it contaminates the food and the water which is there, my dear children. And when we have this food and the water, basically we get infected. Now, houseflies acts as carriers. They are also acting as a carrier. Why? Basically, housefly will be sitting on probably some fecus and then sitting on the food which is there. Okay, now obviously it has transmitted the whole thing over here. So it is going to act like a carrier. It is going to act like a carrier. Remember, it's not inside the body, which is why I'll just call it a carrier, not a vector. It is just a carrier. Now, spreading infection from the fecus to the food. So this is something again that is caused just like typhoid by the contaminated food and water. The feco-oral route. We call it feco-oral route. So from the feces to the oral route, it is getting transmitted. Okay, housefly is a carrier. Amoebiasis, amoebic dysentery, and all this thing. Very, very important, and Amoeba histolytica. Done. So these are the two protozoan diseases. Until now we have studied bacterial diseases, we have studied protozoan diseases. Now it's time for helminthic diseases. What do you mean by helminthic? You know, as helminth, okay, that is a phylum which is there, and usually we call them roundworms. So in helminthic diseases, again you have two things. One is Ascariasis and the other one is filariasis. So Ascariasis is caused by Ascaris, which is Ascaris lumbricoides, my dear children. Okay. Now symptoms, what do you have in symptoms? Again, it is something that affects your intestine. It is a very, very important point. So internal bleeding will be there, muscle pain, fever, anemia, intestinal blockage. So intestinal blockage is something very famous and it is associated with Ascariasis, my dear children. So you might have seen certain pictures, which is a very gross picture, where the worms kind of infect the whole intestine. It is just filled like noodles. It sounds gross, obviously it looks gross as well, which is why I didn't include it over here, but I'm just telling you. So it causes intestinal blockage, my dear children. That is one very important symptom. Apart from that, internal bleeding is there. Okay, internal bleeding is there, that is different from blood and mucus which is there in amoebiasis. Cool. Now how is it transmitted? So eggs excreted in the fecus contaminate soil and water and the plants. Okay, infection spreads from contaminated water, vegetables and the fruits. Same thing, contaminated water and the food which is there, okay, which is vegetable and fruits. Why vegetable and fruits? Basically this water which is there, this water, the contaminated, for example, there is fecus, contaminated fecus being put into the soil or the water. From the water it will be seeping into the plants and everything. We are going to eat these plants. On the surface of it there might be these things, which is why we have said that we should be probably washing it properly, we should be boiling it. So the eggs will be there, the eggs, my dear children, will be there. Now if the eggs enter our body, obviously they are going to cause a huge ruckus over there. Please remember, the eggs are excreted in the fecus, contaminated soil. So the worms which are there, it will be releasing certain eggs. Now this eggs will be moving out of our body in the form of fecus. Now the fecus is mixed with water or basically food or the plants, whatever is there. If you are eating it, we get the eggs back, and basically someone else get the eggs, and basically in their body also the same thing repeats. Okay, so I hope this is also clear. Now the next helminthic disease is filariasis. You also call it elephantiasis. So you can associate this particular disease with the diagram that is given over here. So over here, as you can see, the lower limb which is there, it is kind of swollen. Why, my dear children? Because you know what, in our body there is lymphatic vessels. Okay, the lymphatic vessels job is to carry the extra fluid which is being moved out. So in our body nothing is perfect. So when the blood moves around the capillaries and everything, what happens is that some plasma and everything moves out of the capillaries because it has small pores. Now there is lymphatic vessels which will be carrying all this fluid and taking back. Okay, so that does not accumulate over there. But what if the lymphatic vessels itself are affected? If they are affected, obviously the fluid will be there. So which is why there will be inflammation. So the most common symptom that you have to remember over here is that there will be chronic inflammation, swollen lower limbs, and also there will be genital deformities. So over in the genital areas also there will be deformities that will be caused because of this. So it is the site of infection is what? Lymphatic vessels, my dear children, mainly of the lower limbs. Lymphatic vessels mainly of the lower limb. Now, who is the person who causes this? It is Vaccaria bancrofti or Vaccaria malai. They are known as filarial worms, which is why the disease is filariasis. Okay, or you call it elephantiasis because you have an elephant shaped leg, okay, that much big leg. Now, mode of transmission, how is it transmitted? So, unlike transmission of all the other things that we studied now, it is again caused by a vector, that is basically the female mosquito bite. What female? It is Culex mosquito. Remember the name of this mosquito, please write it somewhere, Culex mosquito. So, it is spread by the mosquito bite, that is female mosquito bite, that is Culex. Now, progression, worm live for years causing gradual swelling and deformity. So, this is a slow change that you observe over there. Okay, so mode of transmission is by the bite of, always remember, mosquitoes are going to be females. The one that affect is going to be a female because female is one of the one that feeds on the blood. Male, they feed on the plant sap. Obviously, it is not going to affect us. So here it is Culex mosquito. There it was Anopheles mosquito in malaria. Okay, I hope this is also clear. So helminthic diseases is done. Now we have fungal diseases. In a fungal disease, there is only one disease being mentioned, and that is ringworm disease. So ringworm disease is not caused by worm, please remember. It forms ring-shaped structure, which is why it is known as ringworm disease. It's a fungal disease which is there. So there will be some swelling over here, not swelling, it will be if you touch it. Okay, if you touch it, basically it will be like a bit upwards, above your skin level. Okay, now it is caused by three types of fungi which is there, microsporum, trichophyton and epidermophyton. So you can remember the outer thing is known as epidermis, right? So on epidermis there will be small hairs, you call it trichomes. So basically on the epidermis there is fight happening. So trichophyton and epidermophyton and also microsporum which is there. They are all fungi, my dear children, and the disease name is ringworm. So what do you have as a symptom? There will be dry scaly lesions on the skin, nails, scalp with intense itching. You will be having so much itching. Now how is it spread? This is spread by contact with infected soil, towels, clothes and combs, which is why we are not recommended to use anyone else's things, right? Now the most favorable condition is heat and moisture. So if there is heat and moisture, obviously there are chances that there you will be getting the ringworm disease or the fungal disease which is there. Okay, now for example you have groin area such as where your what do you call it, suppose this is your lower area. Okay, so these areas there are higher chances that you can get the fungal disease because there are heat, there is moisture, and it is the most favorable condition for them. So there is only one fungal disease which you have to remember, and that is ringworm disease. I hope you do not forget it. Okay, chalo. Let's go ahead. And now we have viral disease. In viral disease, my dear children, you have common cold as the first thing that is given in your NCERT. Okay, so just after pneumonia, the common cold thing is there. So I just shifted a bit forward because that's how you can relate and learn. Okay, so common cold, my dear children, it is caused by rhinoviruses, a group of viruses, which is what? Rhinoviruses. And you know what? Why we cannot treat common cold? Because viruses have a good ability to change the genetic material every now and then. So we find basically a drug, and then they will be changing their form overall. So basically that drug would not be useful for them. That is why we do not have a specific medicine for common cold which is there. So if someone has common cold, it might be staying there for almost like 3 to 7 days until our body totally fights it. Okay, so common cold is there. Now it causes nasal congestion, the sore throat, the cough, the headache and tiredness. I already told you common cold is not going to affect the lower respiratory system. It is going to affect the upper respiratory tract which is there. So nasal congestion, there is sore throat, there is cough, there is headache and tiredness. So in pneumonia you can get both. Pneumonia, from here to here, overall you will be affected. But in the case of common cold, so it's basically this area that is mostly affected. So nose and the respiratory passage, but not the lungs. I'm mentioning again, it's not the lungs that is affected. Mode of transmission, just like pneumonia, inhalation of droplets, contact with contaminated objects like pens, book, key, everything which is there. So which is why if you're sneezing, please wash your hands and touch, because others might also get it. So apart from common cold, which is caused by rhinoviruses, there are other viral diseases, that is dengue and chikungunya as well. Dengue and chikungunya, my dear children, very important. Why? Because it is again caused by a mosquito, by a mosquito bite, that is Aedes mosquito. That is what? Aedes mosquito. In Aedes mosquito, there will be what? The virus which is there, and obviously that's how it spreads. So Aedes mosquito, the female one, obviously it causes dengue and the chikungunya both. So we learned 3 mosquito vectors. I'm going to show you that in the next page probably, but this is what I have for viral disease. Apart from that, you have HIV/AIDS, which is again caused by a virus, that is human immunodeficiency virus. Cool. I hope this is sorted now. Now going ahead, my dear children, what do I have? So these type of questions can come. Which of the following vector-borne diseases caused by Aedes mosquito? So this is dengue and chikungunya. We know it now. Okay. So please, you might not prioritize it because it is just written in one line, which is why I am exaggerating and telling you this. Now over here, see, malaria is caused by female Anopheles mosquito, filariasis by female Culex mosquito, dengue and chikungunya by Aedes mosquito. Now, how to prevent the vector-borne diseases? So vector control for all this thing is prevent water stagnation, because that is where they lay their eggs and stuff like that. Clean the coolers regularly, that is also a breeding ground for them. Use mosquito nets and wire mesh. Then introduce Gambusia fish, because Gambusia fish basically feeds on the larvae of all the mosquito and things like that. Spray insecticides in the breeding areas. Now Gambusia is important, please remember it. Even the questions have not come, but we cannot predict, anything can come, right? So this is about the diseases that we have. Now we are going to talk about the immunity, lots of the diseases, right? Now we have to see how our body is going to fight against it. So my dear children, we have 2 types of immunity. One is innate and the other one is acquired immunity. Acquired as in you are going to acquire it throughout your lifetime. Innate as in it is there from birth. It is there from birth. So it is non-specific type of defense. It is not specific that I am going to act on this particular pathogen or things like that. That knowledge would come slowly to our body, and that comes under acquired immunity. So this is non-specific type of immunity where it is going to be same for all. For example, the skin. Don't we all have skin from birth? Is there someone who has skin later in their life? No, we all are born with a skin. So skin is a type of immunity. It is causing protection. It's giving us protection from anything that try to enter our body. And it is non-specific. It does not say that, hey, virus you can get in, but the bacteria you are not allowed to get in. Is it like that? No, it is common for all. Right, so it is non-specific type of defense which is there. Now there are different types. One is physical barriers. Skin, it prevents the microbial entry. Mucus, which is there in our respiratory tract, in our gastrointestinal tract, in the urogenital tract. All of them, they are responsible for trapping what? The microbes. That is physical barrier. Second is physiological barrier, as in they are releasing something and that is going to help us. So stomach acid, the saliva, the tears, all of them, they are going to prevent the microbial growth, because all of it has something, some enzymes. So for example, in saliva and tears, we have lysozyme, and lysozyme basically kills the breaks open the cell wall of the bacteria and stuff like that, which is why they won't be able to survive. In stomach acid, that is HCl. Can anyone survive? No, my dear children. So it is acting as a physiological barrier. Now you have cellular barriers such as polymorphonuclear leukocytes, as in there will be multiple nucleus and the shape is kind of very much different. So it is why morphology is different. Nuclear leukocyte, that is PMNL, neutrophils. The cells are known as neutrophils, my dear children. Neutrophils and monocytes are there. They destroy the microbes from the very beginning. We have it all, okay, from the very beginning, neutrophils and the monocytes. Apart from that, we have natural killer cells as well in the blood that attack what? Infected cells. They attack the infected cells. And now you have macrophages in the tissues perform phagocytosis. What do you mean by phagocytosis? For example, I have a pathogen, I eat it in a whole. I take it inside and then I break it off. So macrophages, neutrophils, monocytes, and then natural killer cells, all of them, they act as a cellular barrier which is there in our body, okay, from the beginning. Now you have cytokine barriers. What do you mean by that? Basically what happens, if a cell is affected by virus, okay, that cell will be releasing certain things outside, such as interferons, okay, such as interferons. Now what does this interferon do? It goes and tells the other cell, hey, you have to be careful, there is a virus attack in our body, you have to be really careful, be prepared for that. So that is the role of interferons, okay, and it is a cytokine because it is being released from a cell. So it is a cytokine barrier. So virus infected cells release interferons to protect body from healthy cells. So all of them, they come under innate immunity: physical barrier, physiological barrier, cellular barrier, and the cytokine barriers. So over here, if you have noticed, so these are neutrophils, monocytes, natural killer cells, macrophages, all of them, they are kind of different types of WBCs, white blood cells. Now over here, there is one more type, that is what your lymphocytes and all. Lymphocytes are not being mentioned over here because that is not a part of innate immunity. You develop lymphocytes with time after you are born, so it's an acquired form of immunity, okay. So this is all about innate immunity. Please remember interferons, very important thing, and also PMNL and the names of these cells. Cool? Works? Chalo. So they can give you skin, mucus, this. Which one of this is a part of innate immunity? So you should know what is a part of innate immunity. So it is there from birth and it is non-specific. Now acquired immunity, my dear children, it is very much pathogen specific. It is specifically released for specific pathogens. Now it has memory as well. So whenever we get some disease, if you have seen, the first time we are affected, the response is basically we will be having the disease, we will be very tired, we will get fever and everything. So the body is showing a primary response. Our body is showing a primary response, that is in the first exposure, and it will be of low intensity. The response is of low intensity, which is why we get affected. But the second time the same things comes in, okay, the secondary response or anamnestic response will be stronger and faster because in our body there are memory cells being produced. So whenever we get affected by something, our body will be producing certain memory cells which will remember that this is something that had been come, and this is the antibody, or this is the cells that I have released in response to this particular disease, and probably have to increase their count when next time the attack happens. Okay, so it has memory and it is pathogen specific, my dear children. The primary response, it is usually of low intensity. The secondary or anamnestic response, it is stronger and faster because there are memory cells being produced. Okay. Now if we talk about lymphocytes which are being involved, okay, the lymphocytes which are being, here lymphocytes are there. Lymphocytes is again a form of WBCs which are there, okay, and there are B lymphocyte and T lymphocytes. So B lymphocytes are the one that produces antibodies. B cells are the one that produces what? Antibodies, my dear children. In antibodies, you have different type of antibodies. You can remember like this: GAMED, antibody G, antibody A, antibody M, antibody E, and antibody D. Okay, or you call it immunoglobulin. So there are proteins, which is why it is known as immunoglobulin G, immunoglobulin A, M, E, D. So IgA, IgM stands for immunoglobulin. Okay, now whenever we say lymphocytes are being, B lymphocytes are being there, we say it's a part of humoral immunity. So acquired immunity is divided into two parts: humoral and cell mediated. Humoral immunity, because why, my dear children, these cells, B cells, are there in our humor, as in our body fluids, such as in the blood. Okay, so whatever contaminants come in our blood, they are being trapped with the help of B lymphocytes. They are going to produce antibodies, they are going to produce memory cells, and these memory cells are going to be there to remember all the things that happened this time. So next time it will be really fast. Now there are T lymphocytes as well produced in our body, and what does T lymphocytes do? They are going to help the B cells. They are going to help the B cells. So B cells cannot obviously work alone. So who is going to give the signals to the B cell that hey, you have to release antibody and everything? It's the T lymphocytes which does it. It's a T lymphocyte which does it. So T lymphocytes basically are cells, and they are going to release certain things. They're going to make other cells say that hey, there's something happening. Please ask the B cell to produce antibodies and stuff like that. Okay, so it is showing cell mediated immunity. It is going to show cell mediated immunity. In T lymphocyte also you have different type of cells, but still I'm just talking about in general T lymphocytes. Okay, so lymphocytes which are involved is B lymphocyte and T lymphocytes. Whenever we say B lymphocytes are involved, we are talking about humoral immunity, because B lymphocytes are found in the humus or humor, that is the body fluids and everywhere, where they produce what? The antibodies. T lymphocytes, we call it cell mediated immunity because it involves so many cells. It's like a chain reaction. When one T cell is activated, basically it is going to inform other cell. It is going to infect it is going to inform the B lymphocytes and everything. That's how it works. Okay, we need T lymphocytes, a very important thing in our body. Now, we spoke about B lymphocytes and they produce antibodies, right? So, structure of antibodies is equally important. Over here, they have two heavy chains that you can see, and these two are the light chains. So, we denote it as H2L2. Two heavy chains and two light chains which is there. Now over here, the upper regions of the heavy and the light chain is known as variable region. It will be different for different antibody, GAMAD. Okay, but the lower region from here to here, it will be same. It is known as constant region. It is what? Constant region. Now this is where the antigen is going to come and bind. That is, any pathogen which is there, it is going to come and bind over here. Okay, and where is this? This is present on the cell. This is present on the cell. Now, when an antigen comes and bind, what will happen? This cell is going to release some certain things and probably tell other cells also to be active. These cells will be releasing certain things, killing the antigen in total. It will be killing the antigen in total. That is how things works. Okay, I hope that is clear. So, antibody structure, it is made up of 4 peptide chain, 2 heavy and the 2 light chains. So, over here there is a carboxyl terminal and there is an amino terminal, because this is a protein and proteins are made up of amino acid. So, when amino acids are going to join, so obviously at one point, so amino acid has an amino terminal and a carboxyl terminal. So, obviously when they join end to end, what will happen? At one end there will be carboxyl terminal, at the other end there will be amino terminal. So, that upper end there will be amino and the lower end there is carboxyl. Works? Now, whenever we say organ transplantation is happening in our body, so you might have seen we say that a lot of organ transplantation failed, our body responded. Who is responsible for this? So, every time something comes in our body, we consider it as foreign. It is not our body part, it's someone else's. So, it is obviously considered foreign. Now, when this foreign particle comes in, who is the cells? Who are the cells which play a major role in rejection? T lymphocytes, my dear children. T cells are the ones that majorly causes graft rejection, because they are very active cell. They are the ones that take care of our body really well. So obviously something comes in, even though the body does not know that it is useful, so it will try to reject it. It will try to damage the cells which are out there. So always remember, if you are talking about graft rejection, okay, this is known as graft rejection. So who is responsible for that? T lymphocytes. And what type of immunity is responsible? Since T lymphocyte is there, it is CMI, that is cell mediated immunity, which is responsible for graft rejection. A very important question. Okay, tissue and blood group matching needed to prevent rejection. Obviously the tissue and the blood group, everything has to be matched, then only a person is given the a person is able to do the organ transplantation, but still our body will reject it, because we have T lymphocytes in our body. Okay, so what do we do when organ transplantation is given? We give our body immunosuppressants as well. We give our body immunosuppressants. That means our immune system is going to get suppressed. We do not want our immune system to be active, because if it is active, what will happen? It can reject our the organ which is transplanted in there. Okay, so immunosuppressants require post transplant. So this kind of makes our body weak, and which is why there are high chances that we get other diseases, but still we need the organ, right? For at least our body to understand, at least for some time we have to take immunosuppressant. Basically depends, some people take it for the whole life as well. Okay, so I hope this is done. Acquired immunity, it is pathogen specific, has memory. There is 2 types of response, primary and secondary. Secondary is more rigorous, it is faster, because you have memory cells being produced. Now lymphocytes which are involved, B and T. B are the ones that produces antibodies. T lymphocytes is responsible for cell mediated immunity, B for humoral immunity. Antibody has 2 chains, 2 heavy chains and 2 light chains. Okay. Now there is an antigen binding site as well. Now in organ transplantation, which immunity is going to reject it? It is cell mediated immunity or T lymphocyte, which is why we have been advised to take immunosuppressants. Clear? Done? Cool. So going ahead. Children, very important piece of information. Yes. So if you are a KCET aspirant, okay, if you are seeing, obviously you are a KCET aspirant. So one very important thing is that we have launched 2 things. One is Abhyas KCET test series, and the other one is Lakshya KCET crash course. Listen to me carefully. Both of them are equally important. If you are someone who is there in Bangalore, okay, and you want to teach from you want to learn from the teachers in our campuses, then I will advise you to go for Lakshya KCET crash course. A 21-day course where we will provide you place to stay and all. Or if you are from Bangalore, you can come and go each day. No problem. That is there. Okay, now Abhyas KCET test series is there, where we are just providing the test series. Okay, there will be so many tests, there will be video solution for it, there will be chapter wise one-shot videos and everything, which you can avail for rupees 1499. For how much? Rupees 1499. Now, This becomes important because you can stay at your home and do the practice. So I'll recommend someone to take Abhyas KCET test series. If you are thorough with the chapters, you want to do tests and see, that is also one way of learning new things. Or if you are someone who feels that I'm not very clear with my concepts, then I will recommend you for going for Lakshya KCET crash course. Either way, obviously a bit of your concepts and everything will be cleared, and it's you who have to put a lot of efforts, but it will be really beneficial for you. If you want to know more details, it is given in the description box. Okay, now going ahead. Okay, now active and passive immunity. After that, after all those immunity that we were talking, comes active and passive immunity. So what do you mean by active immunity? So it is developed when the body produces its own antibody. For example, I have got some bacterial disease such as typhoid. My body has never seen it. So my body, what will it do? It will produce antibodies. It will produce antibodies. The first time I get typhoid, I will be really bad because primary response is there, it is going to be slow. The second time I get typhoid, my dear children, my body will fight back it really well because antibodies have already been produced, there are memory cells as well. So this type of immunity which is there, my dear children, which I gained throughout my life, which is known as active immunity, which is where I am making my body only to participate in the immunity. It is, my body is really active over here. Okay, it is going to produce antibodies. So it is developed when the body produces its own antibodies. It is triggered by exposure to antigens. Okay, it is triggered by exposure to antigens. It can be natural infection such as I said, typhoid, or probably with the help of vaccination also. In vaccination, what is happening, my dear children? In vaccination, basically we are giving the disarmed or basically the weakened pathogens. Okay, they are not active in our body. So weakened pathogens are given in our body. What will happen? Our body will be producing antibodies. So first time I get a vaccination, I get fever and things like that because I am making my body release certain antibodies. So it is a bit work for them. But the second time, when they actually am infected with some disease, what will happen? My body will remember, hey, I know that I have to release these antibodies. So it will be faster, which is why vaccination is being done. So that my body prepares itself. You get it? But overall over here, my body is going to do everything, which is why it is known as active immunity. So this is going to be a slow response, but obviously a long lasting protection. Why? Because antibodies are produced. So every time I get a disease, it will be there. The antibodies will be produced. Okay, but slower response because I am making my body do the work. Now passive immunity is when I am giving ready-made antibodies to the body. For example, I am a small baby. I've just taken birth. At this point of time, as I said, lymphocytes are a part of acquired immunity. So B and T lymphocytes to form, it will take some time and everything. But I need protection. I'm a baby right now. So what does the mother do? Mother will breastfeed me. So at this point of time, obviously what will happen? The milk which is there, it will have antibodies. It will have antibody, that is antibody A, IgA. This antibody is transferred from the mother to me and I will be basically having the antibodies, ready-made antibodies. Is my body doing anything? No. My body is just receiving the antibodies, which is why I am safe. But what will happen? It is a short term. It is for only a short term. Why? Because these antibodies are going to get finished. They are going to get degenerated at one particular point, right? So this is short-lived immunity which is there, my dear children, but this is how things work. So active immunity when my body is producing antibodies, passive immunity when I am given ready-made antibodies. It provides immediate but short-term protection. Example, colostrum, the first milk which is there, okay, which is yellow color milk, it is rich in immunoglobulin A antibodies for the newborn protection. Apart from that, before even taking birth, the baby would be receiving immunoglobulin G, IgG, antibody G from the placenta. So antibodies cross from the mother to the fetus through the placenta so that it can get protection. Now antibody injection is there. So it is used for emergency protection. For example, I get a snake bite. Okay, I get a snake bite. This time I cannot wait for my body to produce antibodies against it. I will die. So what if I go to a doctor? What will doctor do? Doctor will give me an antivenom. And what does this antivenom contain? It contains the antibodies. Antibodies which can fight back against it really fast. So again I am giving ready-made antibodies. So you call this as passive immunization. What do you call this? Passive immunization. Remember, vaccination is a type of active immunity. Vaccination is where active immunity is involved. Passive immunization is where passive immunity is being involved. Over here, my body is not going to produce antibodies. I am just going to give antibodies. I hope it's clear. Clear? Please remember, when I say IgG, gamed, right? So G is the first one that is being transferred from mother to the fetus. A is the second one that is transferred through the colostrum. And if you talk about E, IgE is responsible for allergies. So in allergic responses, you have IgE being released. M and D, the function of it is not given in NCERT, so you don't have to stress on it. Okay, so this is done. Now we'll talk about vaccination clearly. We'll talk about immunization also clearly. So vaccination, my dear children, I told you, it contains antigenic proteins or weakened or inactivated pathogens. They're not active pathogens. If they're active, we'll get the disease, right? So what do we do? Some things which are there, something that is not of our body, we're just going to insert it into our body so that our body realizes this is foreign particle and it will release antibodies. First time we'll get a bit weakened and everything, but it is going to produce memory cells as well. So it is going to produce antibodies, it is also going to produce memory cells. These memory cells are there, so next time I am going to get infected naturally, what will happen? Basically my body will fight back against it. So it triggers antibody production for future protection. Memory B and T cells enable a faster response on re-exposure. So both B lymphocytes and T lymphocytes are going to produce what? Memory cells as well. So B lymphocytes produces antibodies and also memory cells. T lymphocytes also similar way, they are going to produce memory cells as well for a faster response the next time. Now passive immunization is preformed antibodies are injected for immediate protection. For example, tetanus antitoxin, a very important example. Snake bite antivenom. So whenever we get scratched by iron rod and things like that, people advise us to take a tetanus shot. Why? Because immediately we are given what? Preformed antibodies, my dear children, and this will neutralize the toxins released by the bacteria, which is going to cause what? Tetanus. Now snake bite, again it is snake bite antivenom, it again contains antibodies, again I am giving ready-made things. Fine. So this is about vaccination and passive immunization. Remember, vaccination is a part of active immunity. It induces active immunity in us. Passive immunization induces passive immunity in us. Now, recombinant DNA technology. Why am I telling about this? Because in your NCERT there is a line given and this line is very, very important. So, produces antigenic proteins in bacteria yeast for vaccines. So, vaccines have to be made. That is, the antigenic proteins or weakened inactivated pathogens have to be made. So, how do we make it? So what do we do is that, for example, I want hepatitis B vaccine. So the problem causing, for example, I have some proteins on the hepatitis B, contaminant. Over there, I am going to take the gene that is going to make those proteins and I am going to insert that gene inside yeast and everything, inside the yeast. Now yeast will be replicating. Yeast will be what? Replicating again and again. So I am going to get that particular gene again and again. So that means the gene, whatever protein it codes for, that also I am going to get. Now these proteins can be used as vaccine. This protein is not going to cause trouble in us, but just going to elicit an immune response in us, and it can be taken down for vaccination. So recombinant DNA technology is really, really important for creation of what? Vaccines, my dear children. And hepatitis B vaccine was made from the yeast cells. Please remember this information, from the yeast cells. So the thing that can elicit an immune response for hepatitis B, I have taken that and I have put it in the yeast, I made it replicate and I got the product, I used as vaccine and I gave to people. That's how their body came to know that there is something that could actually come and affect them. So I have to produce memory cells, I have to produce antibodies, I have to be prepared. Cool? Please remember this thing. Hepatitis B vaccine from yeast. Now going ahead with the next thing. Where is it? Yes, children, I hope you're getting it. So I'm going a bit fast because if we go wrong, KCET is near. We cannot take in so much time for explaining one particular chapter. Okay, so yes, next, allergy. So allergy, what do you mean by that? It is an exaggerated immune response. A normal immune response is not like that. But when our body's immune response is doing an extra work over there, which we actually do not need, that is what do you mean by allergy, which is why we start. What says we have swelling, we have runny nose, we have teary eyes and everything. So exaggerated immune response to environmental antigens, that is allergens, is known as allergy. Now common allergens are dust mites, pollen, animal dander, etc. Symptoms, you have watery eyes, you have runny nose, you have breathing difficulty, everything. The cause, why is it caused? Release of histamine and serotonin from the mast cells. So listen to me carefully. What happens is that in our body, we have IgE being produced, antibody E being produced because of the all type of immunity, cell mediated, this and that, and humoral immunity. B lymphocytes are involved, T lymphocytes are involved. At last, IgE is being produced. Okay, at last, IgE, for example, suppose I have some dust in my body. Through my nostrils and things, it entered. What will happen? My B and T lymphocyte, everyone will get active? Okay, B, T, everyone gets active. T cells, the B cells to produce antibodies. What antibodies? IgE antibodies. Now this IgE antibodies will go and bind on certain cells which are known as mast cells, my dear children. Now what happens when these guys comes and bind and there is an antigen attached to it? The mast cells is going to release two things. One is histamine and the second one is serotonin. What does this histamine do? Basically, it is going to make the blood vessels dilated. So suppose the blood vessel was like this, it will dilate the blood vessels. Why? Because it wants more and more cells to come and reach this particular place to fight against the disease. But this is an over exaggerated response, my dear children. And what happens when this happens? Basically, swelling happens when there is vasodilation and everything. There will be swelling, there will be a lot of things. There is unwanted fight that is happening inside our body. So please remember who all are involved. IgE is involved, mast cells are involved, and what is mast cell going to do? It is going to produce histamine and serotonin. Okay, please, allergy, IgE. Now diagnosis, small dose of allergens are tested on the patient. How do you diagnose it, that whether you have allergy or not? You put certain dose of things in your body and test it, very little, okay? Now, usually, for example, I went to a doctor once, okay. So, I had this very bad cold and stuff like that. So, it can be caused by a virus, it can be anything else, right? So, basically, I went to a doctor and then the doctor tested my immunoglobulin E count and it was way too high. That means probably I was having allergy to something. That's how I realized I have some allergy to certain thing which is there. Okay, now the treatment for this was what? Antihistamine, adrenaline and steroids which is there. So antihistamine, as in it produces histamine, to make it less we have antihistamine. So you know this cetirizine is something that is given in allergy because it is having antihistamine properties. If you just turn over and see through the ingredients and stuff like that, you will be able to see this information, that is it contains antihistamine over there. Okay, so please remember antihistamine, adrenaline and steroids, majorly antihistamine which is given as what? As a mechanism to basically treat allergies. Cool. Now next thing, we have autoimmune diseases. So autoimmune disease is like when our body is kind of going crazy. What is going to happen is that our body usually senses certain things, okay, that it is foreign, it is not and things like that. But sometimes our own cells, it is not able to sense our own cells. It feels like our own cell is a foreign particle, and when it feels like that, it is going to damage it. If it damages, my dear children, what will happen? We are going to have a major loss in our body, right? So certain diseases are there and it is known as autoimmune diseases. So when the body mistakenly attacks its own cell, we say that the body is having an autoimmune disease. It can be due to genetic or certain unknown factors which are there, because usually our body has the ability to understand what is ours and what is not ours. Example for this is rheumatoid arthritis. It causes joint inflammation, my dear children. It is going to cause joint inflammation. So rheumatoid arthritis is when the synovial fluid which is there in between the bones and all, we have synovial fluid. So those particular areas are going to get affected. Okay, those cells are going to get affected, which is why there will be very much pain in the joints and also joint inflammation and things like that. So it is a type of autoimmune disease. Okay, cool, works. So this much is given in NCERT, that much is enough. Now we have lymphoid organs. So I told about all this thing, that B lymphocyte, T lymphocyte, this and that, everything is being produced. But who produces it? Who actually produces it? Where do they actually fight against the antigens? So that's how we are going to talk about lymphoid organs. Okay, so lymphoid organs, my dear children, there are two types. One is primary and the other one is secondary. Primary lymphoid organs are where the cells are going to be produced. They are not going to do distribution. They are not going to act over there. They are just going to be produced. They are going to be antigen sensitive. They are going to be sensitive of the foreign particles. That is why antigen sensitive we call it. Now where are they going to act? They are going to act in the secondary lymphoid organs. They are going to be sent to the secondary lymphoid organs. So that's where they will actually work. So you can think this as primary lymphoid organs as a training ground where they are produced and they are trained, but they do not work. Okay, they have not actually met the antigens over there. Okay, actual training, the actual rigorous training will be given in the secondary lymphoid organs where they will meet the antigens. So this is like the theoretical explanation of this, and this is the actual practical thing that happens over here. So here the theory and everything is taught that you have to be antigen sensitive, you have to kill as this and that. This is where actually it works like that. So primary lymphoid organs, there are two. Okay, one is bone marrow and the other one is thymus. So listen to this. Development and maturation of antigen sensitive lymphocytes happen here in the primary lymphoid organs. There are two, that is bone marrow and the thymus. Bone marrow is there inside our bones. In our bone, we have red marrow and we have yellow marrow. Okay, yellow marrow gives rise to fat and everything. Red marrow, my dear children, has lots of stem cells. It produces what? Stem cells. You call them hematopoietic stem cells. As in all kind of cells are being produced from these stem cells. They have the potency to give rise to all different type of cells. That is RBC, WBC, platelets, everything. So in the bone marrow, it produces all the blood cells, including antigen sensitive lymphocytes. It produces that also. Okay, that is B lymphocyte, T lymphocyte, everything is being produced there. The B lymphocyte which is there, it produces, it is what matures here only. In the bone marrow itself, it will be maturing. What about T lymphocyte, ma'am? T lymphocyte will go to thymus for maturation. You know, thymus is behind our sternum. Sternum is the chest bone which is there. Just behind that, you have thymus, my dear children. And what happens in the thymus? In the thymus, the maturation of T lymphocytes happen, which is why T stands for thymus over here. And it shrinks with age. The shape will be shrinking with age. Okay. Hope you got it. So bone marrow is where hematopoietic stem cells are there. They are going to give rise to RBC, WBC and platelets. In white blood cells, you have B lymphocytes, T lymphocytes and all the other cells. Let's just talk about B and T. So B lymphocytes are going to mature in the bone marrow itself. T lymphocytes are going to go to the thymus and mature there. But when are they going to actually face the the antigens? In the secondary lymphoid organs. So lymphocytes encounter antigens and they proliferate, they increase the number over there. So spleen is one such place. Lymph node is one such place. Tonsils, Peyer's patch, appendix is one such place. Malt as a mucosa associated lymphoid tissue is one such place. So basically listen to me. Spleen is one. Spleen is the thing that filters our blood. Okay, spleen is the organ that is there above our stomach. Okay, so what it does is that it's known as graveyard of RBC, because this is where the old and worn out RBC will come, and then it is chosen and it's been degenerated, and the iron and everything is taken, rest is being degenerated. Okay, so that's how spleen works. So this particular time in the spleen, in the blood vessels of the spleen, you have so many lymphocytes as well. You have so many lymphocytes. So whenever blood comes in there and you see a pathogen, these lymphocytes are going to break down these pathogens as well. They will kill these pathogens as well. So spleen obviously is a site where the practical thing works. Lymph nodes. I told you there are lymphatic vessels. I told you there is lymphatic vessels which will be taking in all the extra fluids and everything. So these lymphatic vessels, they have swollen regions like this, and these regions are known as lymph nodes. Lymph nodes have high amount of lymphocytes. So whenever something comes, for example, the fluid which is there, if at all it carries some pathogens, so lymphocytes which are there were going to kill those pathogens as well. So that's why doctor touches here and see whether you have swelling. So here and all you have so many lymph nodes, my dear children, and if it's swollen, that means you are having an infection in your body. So traps and activates antigen sensitive lymphocytes. Now tonsils, Peyer's patch. Now tonsils over here. Peyer's patch which is there, it is now in our intestine, the inner layer of intestine. Appendix which is there, all of them they are site for immune response. Now you have mucosa associated lymphoid tissue. Okay, mucosa associated lymphoid tissue. Now I'll tell you, tonsils and Peyer's patch, everything is a part of this. What do you mean by mucosa associated lymphoid tissue? You know, the innermost layer of our gastrointestinal tract, it is known as mucosa, the innermost layer. Now, in the mucosa, whatever lymphoid tissues are associated, you call them mucosa associated lymphoid tissue. So 50% of the lymphoid tissue is this only. Okay, 50% of the whole lymphoid tissue is malt. Okay, in that you have tonsils, you have Peyer's patches and everything. So it is there in our respiratory, digestive and urogenital tract. So as I told you, tonsils is there over here in the respiratory tract. Digestive and urogenital tract over there, as I told you. Again in the small intestine, what do you have? You have the Peyer's patch. So all of this becomes a part of malt only. And malt is taking 50% of the lymphoid tissue, my dear children. So I hope you remember it. Again the question can come, which among them are primary lymphoid organs, which among them are secondary lymphoid organs. Done? Cool? So that's about lymphoid system. Now we talk about the disease. HIV, very, very important disease. So HIV is caused by human immunodeficiency virus. What is it called by? Human immunodeficiency virus, my dear children, which is a retrovirus, and it has RNA as a genome, as a genetic material. What does it have? RNA. What is our genetic material? Our genetic material is DNA. So it has RNA as a genetic material, my dear children. And also one more very important thing, it is covered, enclosed in a protein envelope. So there is a protein envelope, okay, there's a protein envelope in which the RNA is being covered. Okay, Cool. Now, AIDS stands for Acquired Immunodeficiency Syndrome. The name of the disease is AIDS. It is Acquired Immunodeficiency Syndrome. You acquire it during your lifetime, yeah, and it makes your immune system deficient, which is why you will be having certain symptoms. So, group of symptoms is known as syndrome. So, AIDS definition, a deficiency of the immune system acquired during life. It is not congenital, that means it is not genetic. Now, syndrome, a group of symptoms associated with the immune failure. That is what you mean by syndrome. Now, first reported in 1981, has since caused 25+ million deaths globally, which is totally scary, right? Now, HIV/AIDS does not spread by touch or casual physical contact. Please remember, if I touch you, if I have casual physical contact, there is no chance that anyone can get HIV/AIDS. It is only transmitted through body fluids. Now, this body fluids can be genital fluids, can be blood and everything else. Okay? Please remember that. Works. Now, HIV transmission mode. First is sexual contact. So, there is a question, which among them is not a mode of transmission of HIV? So, you should know, mere touch is not. You get it? So, sexual contact with an infected person, that is because you are exchanging the genital fluids. Okay. Now, we have transfusion of contaminated blood or the blood products. So, obviously, if blood is getting transported, obviously, it will be having. Now, sharing infected needles. So, what happens? The drug abusers and people what do they do? They have the intravenous injections. Now, the same needle is used for another person's entry. So, your blood is there on the needle, my dear children, even though it's not very much visible. So, still you will be having it. Okay. So, mother to child transmission through the placenta only. Through the placenta, there is a chance that it can happen. Okay. There is a chance. Not always. Now, sexual contact, for sure, transfusion of contaminated blood or blood products. So, people who have diseases where you need blood transfusion repeatedly, you have to be really careful. Sharing infected needles and mother to child transmission. Now, high risk group, people with multiple sexual partners, because sexual contact can cause it. Drug addicts, because they are using this. Okay, individuals needing repeated blood transfusion, for example, people who have thalassemia and things like that. Now, children born to HIV infected mothers. So, all of them, they are high risk groups, my dear children. They are high risk groups. Also, one more thing, in sexual contact, there is only one way to prevent HIV, that is what? Using condoms. Using condom, because condom is going to act like a physical barrier over there. So, that's the only one method that can be used as a contraceptive method, and also it prevents the transfer of sexually transmitted diseases. But again, risk is always associated. Cool? Works. Now, HIV attack action plan, how does it actually work? So, HIV infection and I'll tell you. So, the virus enters our body, and there are certain cells which are known as macrophages. Okay, they are macrophages. First thing that they do is they enter the macrophage. Slowly. And as I said, they had RNA. They have what? RNA as a genetic material. We have what? DNA. So, they want to use our machinery and replicate. They want to replicate their genetic material. But how they can do? Ours is DNA, theirs is RNA. So, what do they do is they are going to transcribe a DNA out of RNA. So, usually how does transcription works? If you know, the DNA which is there, it is going to help in the formation of RNA, and this is known as transcription. But when you do the opposite, now I will be calling it reverse transcription. So, basically they have their RNA, they are going to make a DNA out of it with the help of an enzyme that they contain, that is known as reverse transcriptase. Reverse transcriptase. Now, what will happen? The viral DNA which is there, it will slowly come and integrate in our DNA. With the help of another enzyme that is known as integrase. So, they have three enzymes. One is reverse transcriptase, second is integrase, with the help of which it is going to integrate into our host genome, into our genome. Now, what will happen? Once it integrates into our genome, normally when our replication happens, so that particular portion of the DNA is also going to be replicated. So, they are going to produce, all the DNA has information as to what to make and things like that. So, they are going to make their own stuff. They are going to make their protein wall, they are going to make their capsule, okay. They are basically going to make the RNA, everything. So, everything is being prepared. So, their whole structure is being prepared. So, don't you think one particular HIV virus which is coming inside, it is using the macrophage as an HIV factory? Because there are so many things which are being produced again and again. So, macrophage are going to act like HIV factory, my dear children. Slowly, with the help of another enzyme, that is permease, it is going to come outside and affect the next group of cells. The next group of cells. I already told you. What is that? What are those cells which play a major role in our immunity? It is helper, T helper lymphocytes, or we call it what? T lymphocytes. So, T lymphocytes, my dear children, we have two types. One is T helper and the other one is T cytotoxic. T lymphocyte, there are two types, T helper and T cytotoxic. As the name suggests, T helper cells are the one that is responsible for helping all the different type of immunity. It will help the B lymphocytes to produce antibodies. It will tell the B lymphocyte to produce antibodies. If the T helper cells are not there, it is going to cause a huge ruckus in your body. So, and the HIV virus knows that. So, what does it do? After being formed inside the macrophage, they are going to affect the T helper cells which are there. Now, HIV enters the T helper cells, replicate there, produces so many viruses there. Now, they are going to infect more T helper cells. Now, when it happens, it is going to affect more and more T helper cells. Obviously, it is going to affect our body. So, decreasing helper cells is going to cause weakened immune system. So, symptom will include fever, diarrhea and weight loss. Sudden weight loss will be there. There will be diarrhea, there will be fever, everything. It is all the opportunistic infections. For example, the things that is waiting for opportunity to get inside our body, all of them will get in our body and affect us. Why? Because our body is now weak. So the bacteria, mycobacterium, it also causes its problem. The viruses, the fungi, the parasites, that is toxoplasma, which usually our body fights back, will now be able to affect our body because we have an very weakened immune system. Okay, so bacteria, that is mycobacterium, viruses, fungi and parasites such as toxoplasma are going to attack our body. So please remember, they are opportunistic infection and they will infect us because we have a weakened immune system. Diagnosis is ELISA, enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay, and the treatment is antiretroviral drugs. So we are given antiretroviral drugs so that they do not replicate inside our body. But this can only prolong life. There is no permanent cure for HIV virus because it's a virus, and I told you viruses have a very good way of changing the genetic material every now and then. So whatever drug we make, they will change themselves, and that's how it causes diseases. Get it? So antiretroviral drugs can be given to prolong the life but cannot cure the disease. Okay, the person would be having it. It is inevitable to cure it as of now, even though we are working on it. Now, ELISA, enzyme-linked immunosorbent assay, works on a principle of antigen-antibody binding. For example, I have a chamber like this and I have the antibodies here. What are these antibodies? I know that these antibodies are the ones that are produced in a person who is having HIV. I know that. Okay, and now what do? I am confused whether this person has HIV or not. I will take their blood. I am going to carry their antigens. If their antigens comes and binds over this, I'll understand basically that this antigens and this antibodies are going to come together only if they are specific to each other, right? If these antibodies were produced in a AIDS patient, obviously these antigens are the one that causes it. Yes. Now, how do I confirm it? I have another antibody. And these antibodies, my dear children, are linked to certain enzymes. They will also come and bind over here. Still, I am not able to view. I am going to put some substance on which these enzymes are going to act and change the color. If the color change is there, depending on the insect, I'll understand that whether this person has HIV, AIDS or not. So it depends on antigen-antibody binding, my dear children. That is a principle for ELISA. We studied in a different chapter, but you have to know it. Okay, so this is how things work. I have a pictorial representation over here from your NCERT. So you might be asked, like there might be some question like what is M, what is C and things like that. So you should know what all the steps mean. So over here, first thing that you have is the retrovirus that is having the viral RNA core. Inside that you have the RNA, right? Outside it you have the viral protein coat. Now this is going to affect the cell. Over here you see the RNA is being inserted, the viral RNA is introduced, the viral DNA is produced by reverse transcriptase enzyme, the first enzyme which is there. Now this viral DNA which is there, it is going to affect the host genome, it is going to enter the host genome. Now new viral RNA is produced from the infected cell because it is going to use our machinery. New viruses are produced because obviously it is going to produce the proteins as well, and then new viruses are going to infect the other cells. This is how things work. Okay, so over here during replication of retrovirus, viral protein is introduced in the host cell? No. Viral RNA is introduced in the host cell? No. Viral DNA is introduced in the host cell? What do you think? Transcriptase enzyme is introduced into the host cell? Tell the answer in the comment section below and I will be checking it out. So basically there will be a question series for all the top 30 questions and stuff like that, but this is how question comes. I'm just here to tell you that. So what you can do is question number 1 you can put question number 1 and tell the answer. During the replication remember, during the replication of retrovirus, viral protein is introduced into the host cell, okay, viral RNA is introduced into the host cell, viral DNA is introduced into the host cell. What is the first thing that is being introduced into the host cell? You have to know about that, okay. You're talking about host cell, you're not talking about host cell genome, please remember. So tell me the answer, put 1 and tell me the answer. Now identify the enzyme that catalyzes the step labeled as M in the given schematic representation. So over here M is given. So what is M? Reverse transcriptase, RNA polymerase, recombinase, ligase. So if you know this is viral RNA, you make a DNA out of it. So it is reverse transcription that is happening. So it has to be reverse transcriptase. So this becomes your answer. You get it? That is why I told you all the small points are really important. You might tend to not take care of it. Okay? Yes. Now, next thing is cancer, my dear children. So, characters of cancer cell. First thing, it has uncontrolled cell growth. So, it keeps on, it has loss of regulation in the cell division. So, it keeps on dividing. So, it is an uncontrolled cell growth. Apart from that, they have lost the contact inhibition. So, what happens normally is that, for example, I am a cell and there is another cell dividing. We both come in contact. We both will stop our division. This is known as contact inhibition. When you are coming in contact, the cell division is inhibited. But in the case of cancer cell, they have lost this property. So, they do not have contact inhibition. Even if I am coming in contact with other cell, I will still divide. So, normal cells stop dividing when in contact with others, but cancer cells do not. Next thing, tumor is being formed. So, there are two types of tumor. One is benign, other one is malignant tumor. Benign tumor as in confined to one location. So, for example, there are a group of cells that keep on dividing. Okay, they keep on dividing, but they are going to stay at one particular location. Now, there are malignant tumors where what is happening? There are rapidly dividing cells and they are going to invade other tissues and they are going to spread around everywhere. So, malignant tumor, the cells of the malignant tumor, my dear children, they are going to show something that is known as metastasis. What is it? Metastasis. This is a property where cancer cells are going to move around our body, different areas of our body. So, malignant tumors are the one that causes what? Cancer. So, I can start with tumor first. Tumor, there are two types, benign and malignant. Benign is where they are just growing uncontrollably, but they are at one particular location. They are not spreading around. Okay, but I call it a tumor, just a tumor. Malignant tumor, I can call it cancer. Why? Because here the cells which are there, they are showing uncontrolled growth. Okay, they are not showing contact inhibition. Okay, and they are also showing something that is known as metastasis. That is a property of cancer cells. That is the third property of cancer cell. Metastasis. They are going to spread around our body via the blood and everything. Okay, that is why we said that cancer is being spread. Okay, so malignant tumor is nothing but cancer. Now, the causes of cancer, there are three causes: physical, chemical and biological causes. So, physical agents are radiations, my dear children, X-rays, gamma rays and UV rays. It causes the DNA damage. So, we can categorize them into non-ionizing radiation and ionizing radiation. Okay, radiation can be non-ionizing and ionizing, such as X-rays, gamma rays and UV rays, that causes DNA damage. If DNA is damaged, my dear children, obviously the normal functioning will be prohibited. Something else it will be doing, right? And it can be cancerous. Now, second is chemical carcinogens. So, tobacco smoke which is there, it is linked to lung cancer. Again, this tobacco smoke which is there, it is going to induce certain changes in our DNA, and if our DNA is changed, we are obviously in a risk of having cancer. Now, biological causes. So, there are certain oncogenic viruses. There are certain viruses which basically what it does, it inserts something in us, and in our body it is going to turn our normal cells into cancer cells. So, I will say the oncogenic viruses, okay, they contain the viral oncogenes. They are having the viral oncogenes. And these oncogenes, when put inside our body, will be causing cancer. Now, this is cellular oncogenes or proto-oncogenes. What are cellular oncogenes or proto-oncogenes, my dear children? Here, the normal gene that can trigger cancer under specific conditions. So, inside our body, there are certain normal genes which functions very normally. They are there in cell regulation and everything. But if at all some mutation happens in them, then short there is form of cancer being formed over there. So, cellular oncogenes as in they are inactivated version, as in they are actively doing something else, but they are not cancerous. But if some changes happens in them, they can cause cancer, and you call them proto-oncogenes as well. Okay, what do you call them? Proto-oncogenes. So, normal genes that trigger cancer under specific condition are known as proto-oncogenes. Works? Cool? Done? Okay. So, cause of cancer, carcinogens, physical radiation, chemical carcinogens are there and biological carcinogens are there. Please remember, oncogenic viruses and proto-oncogenes, these are very, very important. Okay. Now, going ahead, Cancer detection and diagnosis. How do you detect the cancer? So early detection is basically very helpful. It is crucial for the successful treatment. Diagnosis first is biopsy and histopathology. In biopsy, what do? Basically, if there is a lump and everything, you cut a bit of the tissue. That is known as biopsy, when you are basically taking the sample, taking the tissue, okay? And then when you are going to detect it under the microscope and everything, when you are studying it, it is known as histopathology. So biopsy and histopathology, the tissue sample is stained and examined under the microscope. Biopsy means basically removing that, a little bit portion of that is known as biopsy. And testing is, that is known as histopathology. Now you have blood and bone marrow test, detects the increased cell counts, okay, in the blood and the bone marrow. If you see that are uncontrolled cell count which is there, that means there is a cancer, because that is how the cell is going to divide. In cancer only there is uncontrolled growth, right? Now there are imaging techniques, okay. First is radiography, that is using X-ray, which detects the internal cancers, my dear children. There is CT scan, okay, computer tomography. Again, it is going to use X-rays only, but this is going to give us 3D imaging of the internal organs which are there. So there is a damage in the internal organs, we can get to know that. Now there is MRI, which is a safer thing, okay, because over here we are using X-rays, there are ionizing radiations. Now we have non-ionizing radiations as well as magnetic fields, which are used in magnetic resonance imaging, my dear children. Here you will get a precise idea, detection about the cells or the living tissues which are there in our body, if they are showing any growth, okay. This is a safer thing. MRI, computer tomography and X-ray and radiography basically are going to use X-rays. In CT scan you are going to get a 3D image of your internal organs, and in MRI you are going to see your living tissues as well, okay. Now cancer specific antibody tests, that can also be done. So as I told you, in every disease in our body there will be some release of antibodies against it. So if I am knowing that this particular antibodies are produced only in a cancer patient, I can just test whether this particular person who is there has cancer or not. So identify specific cancer and markers which are there, that is one method. Now there is last one, that is molecular biology techniques, that is to detect inherited cancer genes. So some people, you might have seen, if someone has breast cancer, there are high chances the progeny will also have that, because there are maybe certain inherited genes which goes to every single person who is there in their lineage. So it detects the inherited cancer gene, helps in cancer prevention by advising the higher risk individuals to avoid carcinogens. You can tell them that you should be preventing the carcinogens. Please don't smoke because it has tobacco, and tobacco is a chemical carcinogen. You get it? So molecular biology techniques are developing in that way. We are able to detect the cancer genes, but obviously it is kind of pricey. It is still in the verge of formation. It has not been completely discovered like that, okay. So yes, so these are the things that is there for diagnostic methods: biopsy, histopathology, imaging techniques, cancer specific antibody test, and also molecular biology techniques which are there. Cool. Now cancer treatment. So cancer treatment, the common treatment is surgery, where you remove the complete thing. It is done during the initial stage. Now radiation therapy, where lethal irradiation is given to the tumor cells, my dear children, and you are protecting the normal cells. Then there is chemotherapy, where you are using drugs, you are using chemicals to insert in your body and things like that, and it will be specifically going and killing the cancer cells. But you know what, in chemotherapy, the cells which divides very fast, okay, they are the target. So your hair follicles and everything like that, they are something that divides really fast. So which is why in chemotherapy the cancer cells are also killed, and also the hair follicle cells are killed, which is why you go bald. You get it? So may have side effects like hair loss and anemia, because wherever the cells are there which divide really fast, those are going to affect. So hair follicles and the blood cells which are there, they are one such things. So it is going to kill cancer cells, but also other things. Now these are separate things. Now you have combination therapy, where most cancer requires surgery, radiation, chemotherapy, everything together, depending on basically what level you are. Now there is something that is known as immunotherapy. So tumor cells evade the immune system, okay. Obviously tumor cells which are there, it goes on your immune system and causes huge lot of damage over there. So biological response modifiers are given to the body, okay, so that it induces immunity, because you know lots of cells are being lost over here while doing chemotherapy, and apart from that the cancer cells are also causing trouble. So alpha interferons are given. Alpha interferons are basically modifiers, immune system modifiers. They will make your immune system boost up a bit. Remember, this interferon is different from the interferon we were talking there in what do you call it, innate immunity. their interferons are cytokines basically released from your cells and going to tell the other cells to be aware of the virus cells, okay, virus infection that is being caused. Over here alpha interferons are just like that, but yeah, in a different sense. They are response modifiers. They are going to tell our immune system to be a bit strong, and they are going to help our immune system to be stronger like that. Okay, so surgery, radiation therapy and chemotherapy, please remember, and all these things. And with this we are going to complete cancer as well. Now going ahead with the last topic in your NCERT, that is the drugs. So drugs is a very important topic. Yeah, so drugs I can categorize into 3 or basically 4. One is opioids, cannabinoids and coca-alkaloids. So opioids is the first thing that I have. So opioid, all the drugs which are there, they have certain receptors also. Where are these receptors for opioids present? In the central nervous system, that is your brain and spinal thing. Okay, CNS and also your gastrointestinal tract, your GI tract which is there from where your food goes. So receptors for opioids are there on a CNS and also gastrointestinal tract, very important. Now examples of this opioid is morphine as well as heroin. Morphine and heroin. I called heroin as smack also. Okay, heroin is known as smack also, and it is actually a version of morphine. Diacetyl morphine is actually known as heroin. Okay, so where do I get heroin from? So morphine I am not telling where do you get it from. You get it from the plant, that is opium poppy or papaver somniferum. Talk about heroin. So obtained from acetylation of morphine, which is extracted from the latex of poppy plant. So morphine you get it from the latex. Latex as in there will be milk. Okay, there will be milk in the opium, the pods of the opium plant. This milk is being taken. That's how you get morphine. Now what you are going to do? You are going to acetylate. That is acetylation of morphine happens. That's when you get heroin. That's when you get heroin. Okay, the physical properties is what? It is white, it is odorless, it is bitter crystalline compound. It is white in color, it is bitter, it is odorless, does not have any smell, and it is a crystalline compound. How do you intake this thing? By snorting or by injection. By snorting as in taking like this, and or by injection. Cool. The effects, it acts as a depressant, very important point. Heroin is the only thing that is given in NCERT which is going to act like a depressant. It is going to slow down your body functions and everything, and it can lead to addiction, my dear children. It can also lead to addiction. So that is about heroin. What about morphine? So morphine is actually used by doctors and surgeons. Why? Because it is going to act like a sedative and a painkiller. It acts like a sedative and a pain. You make the person sleep, or you lessen the amount of pain in them, and it is very useful in patients who have undergone surgery. But nowadays people are misusing this. Okay, and high dose of morphine can also lead to death. Please remember. So opioids is basically, they have opioid receptors where in the CNS and gastrointestinal tract. Example is morphine and heroin. Heroin is basically diacetyl morphine itself. Where do you get them? You get from the latex of the opium poppy or papaver somniferum. Okay, physical properties: white, odorless, bitter crystalline compound which is there. It is taken by snorting or injection. You also call heroin as smack. Okay, it is a depressant. It slows down your body. Morphine is a sedative and a painkiller. Okay, now this structure which is given in your NCERT, my dear children, it is of a opioid. You remember, like O, it looks like an O. And what is it exactly? Which opioid? Morphine or heroin? It is morphine. It is what? Morphine. And this plant which is there, it is opium poppy. This is the pod which is there. From here you get the latex, the white color fluid. Cool. Now going ahead, cannabinoids. So cannabinoids, they are chemicals that interact with the cannabinoid receptors in the brain. In the brain again. Okay, now obtained from inflorescence of cannabis sativa. The plant, that is cannabis sativa, its stem, its leaf, its roots, everything can be used for cannabinoids. Now forms, there are different forms. Something that you get from the leaf, something you get from this and that. So it is marijuana, hashish, charas and ganja. Okay, it is made from the flower tops, leaves and the raisins which is being released. So different things are being formed from different parts of the plant. Marijuana, hashish, charas and ganja comes under this. Mode of intake is inhalation or oral injection. You can eat it, or basically there is injection as in drink it or eat it, either way. Okay, or then you have inhalation as well. Effect, that is very much important. It affects the cardiovascular system. It affects the cardiovascular system as in your heart, your blood vessels and stuff like that. Please remember that. Okay, heart. So there it was affecting. Opioids basically affect your CNS and gastrointestinal tract because they have the receptors over there. And over here, if you talk about them, it is affecting your cardiovascular system, my dear children, the brain function which is there. So you can see this is a cannabinoid molecule. This is what? A cannabinoid molecule. So if some steps like this comes, understand it is cannabinoid. So for two things, morphine, and for this the image is given. Now this is a leaf of cannabis sativa. This thing that you see is a leaf of cannabis sativa. Okay, cool, done. Chalo. Next thing, you have coca alkaloids. Coca alkaloids, it is derived from the plant Erythroxylum coca, and it is native to South America. Now you might feel, ma'am, what is the use of this? Array, from this question has come, which is that particular drug where you get from America? So it is Erythroxylum coca, which is very native to what? South America. And what does it interfere? Very important question. So this is going to interfere between the dopamine transport. So you know, one neuron will be releasing certain neurotransmitters to the next neuron. That is how things work. Okay, now when it's too much in excess, it goes back also. But over here, it is not allowing to go back, it is going to shut down this thing. So dopamine is going to be released. So basically that person who is taking this particular thing, what will be having? That person will be having euphoria, sense of happiness, and there will be energy and things like that. And if high doses it is taken, it can cause hallucination. I can see people flying and things like that. You get it? Even though it sounds fun, but it's not fun, it is very dangerous. Because you seeing people fly, you also try to fly, you might fall down. And it's not, literally, I'm not joking around this thing. You might go to the terrace and try to fly, what will happen? You get it? So it's very dangerous. So it interferes with the dopamine transport. This is a neurotransmitter, my dear children. Okay, the common name is coke or crack. Coca alkaloids known as coke or crack. The mode of intake, it is knotted. Okay, and stimulant, it decreases euphoria and energy. High doses, it causes hallucination. Cool. And it affects your CNS, central nervous system. So two things, opioids as well as coca alkaloids affects your CNS. Opioids affects your gastrointestinal tract as well. Over here it just affects your CNS, which is why you have all this thing. Dopamine transport is majorly affected. Euphoria and energy you get hallucination also because of coca alkaloids is native to South America. Okay, you get it from Erythroxylum coca. Okay, cannabinoids are affecting your cardiovascular system, please remember that. Now there are certain other plants also which is having hallucinogenic property, for example Atropa belladonna and datura. So over here the picture is given for datura. So wherever you find a plant like this, okay, it is datura, my dear children, whereas do not get confused with this one. This is totally different. This is kind of very open, so it's open as in opioids. Okay, but this is kind of like this is known as datura. So abuse in sports. Some sports person misuse cannabinoids. About cannabinoids, the other one, cannabinoids. Okay, works. Okay, please remember coke, crack, their opioids are known as smack, and I hope everything else is done. Now, which among the following plants is a source of drug which is native to America? I told you. So it is Erythroxylum coca. I hope you know the answer now. Now, drug abuse. A little small paragraph is given in your NCERT about drug abuse, so we should know about that also. So drug abuse definition is, it is non-medicinal use or excessive intake of drug. Okay, it impairs physical, physiological and psychological function. Medicinal drugs that are abused. So you know, in our system we have barbiturates, we have amphetamines, and we have benzodiazepines. Please remember the name: barbiturates, amphetamines and benzodiazepines. Now this is usually used to treat depression and insomnia. Insomnia as in I'm not able to sleep. So to induce sleep and things like that, we have this particular thing. But people are abusing it, people are overusing it, and yes, that causes the trouble. So barbiturates, amphetamines, benzodiazepines used for treating depression and insomnia. Very important question. Morphine, I told you, it is a sedative and painkiller used after surgery but can be misused again. Now hallucinogenic substance, you can find hallucinogenic substance in plants, fruits and seeds also, and they are used in folk medicines and stuff like that. So that is there, but you should not be misusing it. That is something else. So these are very important points. Apart from the other drugs, you have to remember them also. Now, I think we have finished it, but I want to tell you about one more drug that is in the tobacco. It's nicotine. It's what, my dear children? Nicotine. And what does nicotine do? Nicotine, it increases the blood pressure. Nicotine is a very bad thing that increases the blood pressure, my dear children. So make sure no one is getting addicted to this particular thing. So that is also a drug. Okay, and increase in blood pressure and blood levels. Now it also causes release of adrenaline. It's a hormone. You know adrenaline, noradrenaline. So adrenaline basically increases the blood pressure, which is why blood pumping and everything will be really fast. Okay, so nicotine indirectly affects the release of adrenaline, which increases the blood pressure and everything which is there. Okay, and it is also a carcinogen. Apart from that, what I have to tell you, yeah, when smoking is happening at that particular point of time, carbon monoxide is also could be released, and carbon monoxide is a problem with that. So carbon monoxide and oxygen will be competing to be with hemoglobin. So we want oxygen to be with hemoglobin, that's how oxygen will get transported inside our body. But if carbon monoxide is there, it is kind of more more closer to Hb, that is hemoglobin, so it will be carrying carbon monoxide, and our body will be having a deprivation of oxygen as well. So this is a very important problem to be addressed. So please remember nicotine as well, along with all the other drugs which are being mentioned. Barbiturates, amphetamines and benzodiazepines are usually being forgotten, but please don't forget it. And also this thing, that is the datura and atroba belladonna. Cool. Okay, now drug and alcohol abuse, an extra part of it. Now listen, in your NCERT, if you go after drug, this thing, you have alcohol thing, okay, where you're talking about withdrawal syndrome. So I'll just tell you what is withdrawal syndrome. So when a person who is having alcohol regularly, they stop drinking it. What happens is that our body is addicted to it. So our body will be starting to show certain symptoms. We'll have shivers, we'll have things like that, because we need that particular thing over there. This is known as withdrawal symptom. That can be kind of dangerous for certain people, which is why it is not recommended to completely stop anything really fast. In a slow pace it is done, and that's how our body will be adjusted to it. First thing, do not have it. Otherwise your body is going to have lots of trouble. Okay, now there is another thing given in the last pages of your NCERT. Okay, that is about drug and alcohol abuse by sports people. So we have to know about that also, because you get questions from there also. So alcohol use in adolescence can lead to heavy drinking in adulthood, my dear children. And if you have more amount of alcohol and things like that, it damages the nervous system and causes liver cirrhosis as well. It causes liver cirrhosis and damages the nervous system. Now during pregnancy, it harms fetus. Now misuse in sports. So use of narcotic analgesics, as in painkillers, okay, analgesics, anabolic steroids and diuretics, hormones, to enhance the performance. People use narcotic analgesics, anabolic steroids and diuretics or hormones to enhance the performance. But what are the side effects of these anabolic steroids which are being used? So in females, masculinization will happen. They will have masculine body, mood swings will be there, depression will be there, irregular periods will be there, excessive hair and deep voice will be there in the case of females. What about males? Males will have acne, they will have mood swing, depression, reduced testicle size, low sperm count, baldness and prostate issues will be there. In adolescents, severe acne, stunted growth and everything will also be there if they take anabolic steroids. Now this is where most of the questions come from this part. Okay, so anabolic steroids, what are the causes in females? What are the symptoms in females? So masculinization, okay, irregular periods, mood swings, depression, okay, deep voice, excessive hair, just like boys, all the things like boys, you can think like that. Okay, in the case of males, there will be acne, there will be mood swings, there will be depression, reduced testicle size, low sperm count and everything. In adolescents, there will be severe acne and the stunted growth. Premature bone closure will be there. Okay. So question, I'll show you how does it come. One of the side effects are the use of anabolic steroids in females. Cirrhosis of liver is by alcohol. Loss of memory, masculinization is something that is there. Hallucination is because of other drugs. Okay, so this is a 2022 question, so you can expect anything coming around. So I hope I gave you an idea of what type of question comes. So please make sure you are giving attention to all the small details right there. Cool? Works? So that's all, my dear children. I hope it works for you, and do let me know if you have any doubts which are there. I am here to solve those doubts. You can put it down in the comment section. I am going to take that on priority, and wishing you all the best. Lots of love. You are going to crack KCET. Bye bye.